Welcome to Omni Academy. This is Maria, and today we are starting our lecture series on embryology. So, in the upcoming lectures, firstly, we will be studying the development of eye, and in today's lecture, we will be studying firstly the time when this process of development of eye starts in an embryo. Then, we will be studying the sources of development, and then we will be studying the master regulator gene for eye development, and in the end we will be studying the outcomes of development. So let's start with the time. So it is actually the fourth week or the 22nd day of gestation or intrauterine life when this process of development of eye starts in an embryo. This is ultrasound image of a fetus taken at 22nd day of gestation. The presence of optic vesicle in the fetus indicates that now the process of development of eye has begun in the fetus. So now let's move to the sources of development. So we need four primary embryonic sources to develop human eye, and what these sources are: neuroectoderm of diencephalon, surface ectoderm of head, mesoderm between neuroectoderm and surface ectoderm, and neural crus cells. So these are the four sources we need to develop human eye, and in the upcoming lectures we will be seeing that how each source is contributing to the development of different human eye structures. So now let's move to the master regulator gene for eye development. It is PAX6. Any defect in this gene will lead to eye anomalies. So now let's move to the outcomes of development. These are actually the structures that an adult human eye contain. And what these structures are: number one, eyeball; number two, lens; number three, chambers of the eyeball. So let's start with the eyeball first. The four sources which we have discussed collaborate to form the three main layers of the eyeball. So the eyeball has three main layers, and what these layers are: outer fibrous layer, which is composed of sclera and cornea; middle vascular layer, which is composed of choroid, ciliary body, and iris; and the inner retinal layer. So let me explain this with the help of diagram. This is image of a well-developed human eye. This is cornea, which is in the most anterior aspect. As you can see, this is the sclera, which is in the most posterior aspect. Together, they are making up the outer fibrous layer of the eyeball. This is choroid. This is ciliary body, and this is iris. Iris is attached to ciliary body, and iris, ciliary body, and choroid together they are making up the middle vascular layer of the eyeball, which is beneath the outer fibrous layer. You can see in this diagram as well. This is the innermost retinal layer. This is lens. Let me show you another diagram. So this is another diagram of a well-developed eye. You can see that this area, which is in the posterior aspect, this one is the sclera. Sclera in the posterior aspect is continuous with the dura mater of the brain. You can see in this diagram as well. And the area which is present in the anterior aspect, this one is the cornea. So the sclera and the cornea together are making up the outer fibrous layer of the eyeball. So this is the outer fibrous layer of the eyeball. Beneath the outer fibrous layer is present the middle vascular layer. This red layer is the choroid. So this is iris, and this small area is the ciliary body. Iris, ciliary body, and choroid together they are making up the middle vascular layer of the eyeball. So this is the middle vascular layer of the eyeball, which is present beneath the outer fibrous layer, as you can see in this diagram as well. And beneath the middle vascular layer is present the inner retinal layer, and the inner retinal layer is again divided into the pigmented layer and the neural layer. So this black layer is the pigmented layer. It is black in color because the pigmented layer contains a pigment called melanin in it, which is giving the blackish appearance to the pigmented layer. So this is the pigmented layer. And below the pigmented layer, this brown layer is the neural layer. And together the neural layer and the pigmented layer are making up the inner retinal layer of the eyeball. So this is the inner retinal layer of the eyeball, which is present beneath the middle vascular layer, as you can see in this diagram as well. So these three are the layers of the eyeball. So let's now discuss the lens. The lens is made up of primary fibers which are present in the center and the secondary fibers which are present in the periphery. We will be discussing the lens in the development of lens in our upcoming lectures. So let's now move to the chambers of the eyeball. The eyeball has two chambers. The chamber which is present behind the lens, this area is the vitreous chamber and it contains vitreous humor in it. And the chamber which is present in front of the lens, this area is the aqueous chamber. But because of the presence of the iris, the aqueous chamber is again divided into the two chambers, anterior aqueous chamber and the posterior aqueous chamber. Posterior aqueous chamber is between the lens and the iris. So this area is the posterior aqueous chamber. And the anterior aqueous chamber is between the iris and the cornea. This area is the anterior aqueous chamber. So these are the chambers of the eyeball. So this was today's lecture. In the next lecture, we will be studying the early morphogenesis of the eye. See you in next lecture.